What's up? Doc Mike. Public Health on Call. By Johns Hopkins Bloomberg School of Public Health. Today's topic for November 6, 2020. An update on COVID-19 treatments, monoclonal antibodies, convalescent plasma and remdesivir. Thank you, Johns Hopkins Bloomberg School of Public Health.
Welcome to Season 2 of Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health. I'm Joshua Sharfstein, Vice Dean for Public Health Practice and Community Engagement, and a former Secretary of Maryland's Health Department. Our goal is to bring scientific evidence and experience to the public health news of the day through informative interviews with scientists, community leaders, policy experts, public health officials, clinicians, and more. If you have ideas or questions for us to cover, please email us at publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Today, Stephanie Desmond talks to Dr. Arturo Casadevall, the chair of the Department of Molecular Microbiology and Immunology at Johns Hopkins. COVID mortality rates are falling, and they discuss what may be a reason why: better treatments. They cover monoclonal antibodies, convalescent plasma, and remdesivir, as well as what is in the pipeline. Let's listen.
Arturo Casadevall, thanks so much for joining me.
Thank you, Stephanie.
So today I'd like to talk a little bit about treatments for COVID. We are seeing that mortality rates are going down. Are treatments the reason?
Yes, I think that, there are two major reasons that mortality is going down. First, a lot of the people who are getting COVID today are younger, and the mortality in the younger, fortunately, is much lower. But the other thing is that since March 1st, there have been significant advances in how patients are treated, and I'll review some of those with you. Doctors learned that you could improve survival by turning patients over when they were on respirators. I think the two biggest contributors to the declining mortality in the hospital, one of them is that the use of steroids in patients that are intubated has been associated with a reduction in mortality. And the other major change in the United States has been the use of convalescent plasma, that even though its efficacy is still being evaluated, and we cannot say anything definitively, the preliminary indications suggest that it's associated with a reduction in mortality if used early in the hospital course.
Could you give us a little reminder of what convalescent plasma is?
So convalescent plasma is, let's look, what is plasma? Plasma is the liquid that holds your blood together. You know, the red cells, the white cells, the platelets. So when you become ill with an infectious disease and you recover, you make antibodies to whatever made you ill. In this case, it will be SARS-CoV-2. That's what causes COVID. And those antibodies are in the fluid in the blood, and they can be taken out into plasma, which is done by basically people sit down and they have a needle put in their arm, and the liquid is taken out, and that can be used to treat other patients. And the good thing is that a person who recovers can produce enough plasma very easily to treat 2 or 3 other people.
That's terrific. What about remdesivir?
So remdesivir is an antiviral. It's a drug. It interferes with the virus replication. It's just been approved by the FDA, and I think that it is a cause of celebration that something like this could be developed so rapidly. The one thing about remdesivir is it doesn't appear to be that effective. For example, it enhances recovery, it shortens the hospital time, but so far hasn't really been associated with a reduction in mortality. Yet all the doctors, including myself, are very happy to have an antiviral, and it gives us hope that better antivirals are on their way.
Is it sort of similar to Tamiflu? When I get the flu, they give that to me to reduce the severity?
Yes, it's similar in the sense that Tamiflu is also not one of these. The best antivirals that we have, for example, against HIV. Think about it. With HIV, it was a uniformly lethal disease, and now with the antivirals, people can live normal lives. That is what we call great drugs. For flu, we don't have anything like that yet, but it's similar in the sense that if you take the anti-flu medications, you get better quicker.
So I know that you said that the jury's still out on the plasma. I know a study just came out in BMJ suggesting that it has no effect on mortality of COVID patients. What's your thought on that?
Well, the study is inconclusive, and the reason for that is it was used late. If you look at the study, 82% of the patients already had their own antibody. So basically, that was late in the course of infection, and we know that antibody use late is not very effective. And the second thing is that a lot of their units have very little antibody in them. They used donors with mild disease that did not have a lot of antibody responses. So even though it was a great effort, and I commend my Indian colleagues for doing this, unfortunately, it doesn't really help us very much because the study design doesn't address the issues that are important for the use of plasma. But I would point out to you that even though they did not see a reduction in mortality, they saw a reduction in the fact that the patients got better, and also their virus, the plasma helped them clear their virus.
So you mentioned antibodies. So I'd like to talk also about monoclonal antibodies, because I know they're getting a lot of press. President Trump called them a cure because he received a cocktail. Tell me what they are and what role you think they may play.
So when you have convalescent plasma, you have all the antibodies that a person makes. However, it is possible to take cells and to take one of those antibodies and make lots of it. So a monoclonal antibody is a single antibody. Sometimes they put them together, they get what is called a monoclonal antibody cocktail, and that is what the president received. So both convalescent plasma and monoclonal antibodies are what we call antibody-based therapies, but they differ in how they're made. Convalescent plasma is made in people who donate their plasma, and monoclonal antibodies are made in essentially a factory. So the big difference is that every unit of plasma is a little different, whereas monoclonal antibodies tend to be the same. They tend to be standard product, such that every vial is the same.
So if it's being made in a factory, then why is it more expensive than convalescent plasma?
Convalescent plasma is only $300 to $400 a unit, and usually you need only 1 or 2 units. So it's probably the cheapest therapy that we have available. The reason that monoclonal antibodies are so expensive is because they have to be made in what we call tissue culture. You have to grow these cells, and these cells have to produce the protein, and then it needs to be purified. And it's extremely, not only labor intensive, but the reagents are very expensive. So in general, monoclonal antibody therapies, whether for COVID or for cancer or for rheumatological problems, tend to be expensive.
So Trump says he was cured by these monoclonal antibodies. Is that possible?
Well, we will never know the answer to that. What we can say is that he recovered rapidly, and that is consistent with antibody therapies. But we can never know, because it's possible that had he not received his antibodies, he would have still recovered possibly. It is also possible that the reason that he didn't get worse is because he got the antibodies. However, I think most of us believe that he was probably helped by these antibodies, and that probably contributed to his very rapid recovery.
Now, I know that he has called for an emergency use authorization for the monoclonal antibodies, but that hasn't happened yet. Do you see that happening?
So I think it's possible. Plasma is under emergency use authorization. The FDA, based on all the information, allowed it in late summer. And an emergency use authorization is a low bar. It means that it's relatively safe and probably effective. I think that I understand that several companies have already applied to the FDA for such authorization. And based on what we know of plasma, based on what we know of antibody, I think there's a good likelihood that they will be coming.
Mm-hmm. Is it possible to do enough research on these antibodies right now, given that it's hard to make a lot of it and it's very expensive?
So yes, it's possible. The reason, I mean, they're available and they have been given to patients. There are ongoing clinical trials with the monoclonal antibodies. So there is certainly sufficient amount for research, and there is sufficient amount for the clinical trials that are ongoing. I think the issue is not so much a completion of the trials, but what would happen if these reagents were to prove very effective? Could you rapidly scale them up to treat a population where you have millions of people infected? And we don't know that.
Yeah. So there's a lot of excitement, I guess, about the monoclonal antibodies. I've heard them called sort of a bridge to the vaccine. Talk to us a little bit about that.
So I think that is probably not a good terminology, because I think they work very differently. I think when you get a vaccine, you make your own antibodies and then you're immune. I think these antibodies, if they're given to you before you get sick, for example, can give you some immunity, but they don't last very long. So the way I would think about it is they are advanced therapies that are hopefully on the way. And I think we're going to be using convalescent plasma into 2021. And at some point, hopefully, we will have better antibody-based therapies, which will hopefully be either the monoclonals or the hyperimmune globulin that is also under development that is made from plasma.
I was going to ask about that, what the difference is.
A big difference. So when you get convalescent plasma, you get a unit that comes from a patient. However, you could imagine collecting thousands of units and bringing them into essentially a factory where what they do is they take out the antibody and they concentrate it. So then you get only the antibody, and that's known as hyperimmune globulin. That is just began clinical trial.
Okay. Also promising.
Also promising. The advantage over plasma would be that it's more of a standardized product, so that you know what's in each vial, similar to the monoclonal.
So I know that there's a big push right now toward getting a vaccine, and there's questions, I guess, about how effective vaccines would be. I'm curious your thought as someone who's been studying these things for a long time, whether people are going to sort of decide, oh, the vaccine is here, and that means we can stop all these prevention measures, when a vaccine can be approved if it's only, say, 50% effective.
So I think we should probably separate the vaccine efficacy from when we stop in the prevention. I think we'll stop prevention once we reach a certain level of immunity in the population. I think that the good thing is that at least a dozen vaccines are under advanced development. And given that people, when they recover from COVID, mount very strong antibody responses that are neutralizing for the virus, that gives us tremendous hope that some of these vaccines are going to be effective. Now, what does it mean to be effective? Well, we have some vaccines that you have to take only once every 10 years, like, for example, tetanus toxoid. We have other vaccines that immunity doesn't last very long, and you have to get them a lot more frequently. We get vaccinated every year for the flu. That changes every year. So I think the important thing will be to get a vaccine out there that gives protection on a significant amount of the population, even 50% I take. Because if you can immunize 50% of the people, you could significantly prevent transmission, and those people would be protected. But I'm hopeful that the type of vaccines that we've seen in development will give us a lot better than 50%.
Mm-hmm. And you're seeing, in your mind, good antibody responses coming out about the vaccine trials.
I mean, when people recover, it's a funny illness in that the ones who get mild disease don't make a very high antibody response. That's what happened with the donors to the Indian study. But people who get sick and are symptomatic for a while, that is with fever, cough, shortness of breath, those tend to make a very strong antibody response. So our view is that certainly the immune system can handle this. It's not like HIV. It's not like some of the other viruses and microbes that we have trouble with. And given that is the case, if we can reproduce that immunity with a vaccine, and all indications are that we probably will be able to, then we should be able to protect people.
Arturo Casadevall, thank you so much for joining me.
Thank you.
Public Health on Call is produced by Joshua Sharfstein, Lindsay Smith-Rogers, Stephanie Desmond, and Lamari Morales. Audio production by Spencer Greer, Niall Owen McCusker, Cian Oates, and Matthew Martin, with support from Chip Hickey. Distribution by Nick Moran. Thank you for listening.